Welcome to the Deep Dive. Today we are tackling one of the biggest achievements in, well, maybe all, of computational science, solving the protein folding problem.
Yeah.
For decades, this was science's Everest, a computational challenge people thought might be impossible.
It really was the ultimate barrier. And the core problem, it was laid out years ago by Cyrus Levinthal. He basically highlighted the sheer scale of the challenge.
Right.
If you take a pretty small protein, say 100 amino acids, if that protein had to randomly search for its correct 3D shape, it would have to check something like 5 times 10 to the 47th possibilities.
5 with 47 zeros.
Yeah.
It's a number that doesn't mean anything. It's more possibilities than atoms in the universe.
Exactly. It would take longer than the age of the universe. And yet this happens inside our cells, millions of times a second. In milliseconds.
And that's the paradox. It's not random. It's a guided, incredibly fast process.
So our mission for this deep dive is to unpack how AlphaFold, specifically versions 2 and 3, finally crack that code. And maybe more importantly, what you can do with it now that the puzzle is solved.
Right. But before we get into the AI, maybe we should quickly define what a protein actually is. The hardware, so to speak.
Good idea, in the simplest terms.
Think of it like a molecular machine. It's built from a one-dimensional string of instructions. These are the 20 different amino acids. The order of that string, that's the primary structure.
And the whole goal is to predict how that 1D string folds up into its final, unique 3D shape.
Yes, the tertiary structure. Because that shape is everything. It determines the protein's function, whether it's an enzyme or, part of your hair.
And for almost 30 years, the entire field had this brutal competition to measure progress. CASP, the Critical Assessment of Structure Prediction. It started back in '94, and it's basically the Olympics for this field. Teams blindly predict structures that have been solved experimentally but not yet published.
And the winner is decided by a score, the GDT.
The Global Distance Test, yep.
So for the first few decades of CASP, progress was slow, incremental. People were using homology modeling, basically guessing a new structure based on a similar one we already knew.
And then DeepMind shows up. AlphaFold 1 in 2018 was a big step, but it was still kind of in that same paradigm.
But then came CASP 14 in 2020.
That was it. That was the moment. AlphaFold 2 or AF2, it blew everyone away.
Yeah.
It achieved a median GDT score of 92.4.
92.4. And to put that number in context, what does that actually mean?
It means the problem was solved. I mean, a GDT score of 90 is considered to be on par with experimental methods.
So it's as good as getting the structure in a wet lab.
It's within the margin of error of the best physical methods, yeah. This wasn't just an improvement. It was a total phase transition for the field. For single proteins, we could now reliably predict the structure.
What's so amazing is how it did it. It wasn't through brute force physics simulations. It was an insight from evolutionary biology.
Absolutely. The key is that it doesn't predict from just one sequence. It needs what we call a multiple sequence alignment, or an MSA.
So it goes out and searches these massive genetic databases.
Right, for hundreds or thousands of related sequences from other species, bacteria, plants, anything. It's looking for the protein's evolutionary cousins.
And then you stack them all up in this giant table.
Yes. And the genius is in the coevolution theory. The idea is if 2 amino acids at positions, say I and J, are touching each other in the final 3D structure, they have to evolve together. If I mutates, J often has to mutate as well to kind of compensate and keep the structure stable.
So the network just looks for those correlations. If it sees 2 columns in that huge alignment changing together across millions of years of evolution, it assumes they must be physically close.
That's the core insight. It's inferring a 3D contact map just from looking at 1D sequence data. It's brilliant.
And the architecture that pulls this off is the Evoformer, a special kind of transformer.
A masterpiece of geometric deep learning, really, because it has to take all that evolutionary data and somehow force it to make sense in 3D space.
Okay, let's try to unpack this without getting too lost in the weeds.
Yeah.
A normal transformer, like in a language model, just works on a 1D line of text. The Evoformer is different.
It has to handle 2 things at once. So it splits the information into 2 tracks that are constantly talking to each other. You've got the MSA representation, which is the raw sequence data.
Right.
And then critically, you have the pair representation. And this is an L by L matrix, where L is the protein length. It encodes the relationship between every possible pair of amino acids.
So it's like a learnable graph. The model is constantly updating its guess about the distance between every 2 points in the protein.
Exactly. And the Evoformer's job is to update both tracks. It refines the sequence info, and it refines that spatial map back and forth. But there's a problem.
Which is?
A standard attention mechanism doesn't understand geometry. The basic rule that the distance from A to C can't be greater than the distance from A to B plus B to C, the triangle inequality.
Right. The model needs that built-in physical constraint.
And that's the secret sauce, triangle attention. Instead of just looking at the relationship between point I and point J, it updates that relationship by gathering information from all other possible points, K. It checks every possible triangle.
So the geometry is constantly self-correcting. It forces that whole L by L map to be geometrically plausible before it even tries to build the 3D model.
Precisely. Once that abstract map is consistent, the final piece is the structure module. It takes that map and turns it into actual 3D coordinates.
And it does that using something called invariant point attention, IPA.
Yeah, and the key there is what we call SE3 invariance. It just means that if you take the final predicted structure and you spin it around in space,
the internal distances and angles don't change.
right. The model learns to predict the structure in a local reference frame for each little piece, so the global orientation doesn't matter. It ensures the whole thing is physically stable.
Okay, so that's AF2, a revolution for single proteins. But it had a big limitation.
A huge one. It was a specialist. Proteins don't exist in a vacuum. They interact with everything, DNA, RNA, drugs. And trying to get AF2 to predict, say, how a drug binds to a protein, it required all these weird hacks.
Which leads directly to AlphaFold 3. And this feels like a totally different paradigm, a shift from that geometric approach to full-on generative AI.
It is. The first big change was what they called unified tokenization. AF3 just treats everything, amino acids, DNA bases, even the individual atoms of a drug molecule, as part of one long sequence of tokens.
So it can now naturally model the entire interactome. A protein binding to a drug is just one coherent problem for it to solve.
Exactly. But the biggest change, as you said, was the mechanism. It completely ditched the regression approach of AF2 and embraced diffusion models.
This is the same tech behind things like Midjourney creating images from noise.
It's the same fundamental idea. During training, you take a perfect known 3D structure and you slowly add random noise until it's just a meaningless cloud of atoms. The model's only job is to learn how to perfectly reverse that process.
So at inference time, you just give it a cloud of noise.
You give it noise, and it iteratively denoises it step by step until a beautiful, coherent molecule emerges.
Why is that better than the old way?
Because of multimodality. Regression, what AF2 did, tends to predict an average of all possible correct answers. So if a drug can bind in two different ways, a regression model might predict a structure that's an impossible average of the two.
A chemical Frankenstein.
Right. A diffusion model can actually sample and show you both distinct, valid poses. And since molecules in biology are flexible and dynamic, that's a massive advantage.
So to sum it up, AF2 was geometric deep learning, all about the Evoformer and evolutionary data. AF3 is generative AI, all about diffusion and modeling the entire molecular world at once.
That's a great summary.
The moment this technology was released, the impact was just staggering. The AlphaFold database now has, what, over 200 million structures?
It's unbelievable. The experimental database, the PDB, took 50 years to get around 180,000 structures. AFDB gave us 1,000 times more data in a couple of years. We went from a world of structure scarcity to one of structure abundance.
But with all that data, there's the question of trust. If you're a biologist using this, how do you know which parts of the prediction are good?
There are two critical metrics. The first is pLDDT. It's a score from 0 to 100 for every single amino acid, telling you how confident the model is about its local environment.
And the really cool thing is when that score is low.
Right.
Yes.
A low pLDDT score, say below 50, often doesn't mean the model failed. It often means it correctly identified an intrinsically disordered region, or IDR.
A part of the protein that is supposed to be floppy and flexible.
Exactly. The model's basically saying, I'm not sure where this is because nature hasn't decided where it should be. That's an incredibly powerful biological insight.
And the second metric is PAE.
The predicted aligned error. It's a map that tells you the model's confidence in the relative position of any two parts of the protein. If you have two solid domains connected by a flexible linker, the PAE will be low within the domains, but high between them.
So it's telling you, I know the shape of the two pieces, but I have no idea how they're oriented relative to each other. It stops you from over-interpreting the structure.
It's essential for proper interpretation.
Let's talk about some real-world case studies. This isn't just theoretical, it's accelerating actual science.
The malaria vaccine work is a perfect example. For years, researchers were trying to solve the structure of a key parasite protein, TSF4845, but it was a shapeshifter, too flexible to crystallize.
So they couldn't get a clear picture of it.
Right. But AlphaFold predicted that one specific part of it, one domain, was very stable. Based on that prediction, they were able to engineer a smaller, stable version of the protein, which they could crystallize.
Wow.
And that structure is now the basis for a vaccine candidate in clinical trials. The AI didn't replace the experiment, it enabled it.
That's amazing. And what about just pure speed?
Antibiotic resistance. We need to understand these things called efflux pumps, which bacteria use to spit out our drugs. Solving their structures used to take years. A PhD thesis.
Oh, no.
Now you can get a high-quality prediction in 30 minutes. It completely changes the pace of drug design.
And then there's the puzzle-solving approach for huge molecular machines like the nuclear pore complex.
Exactly. The NPC is massive. Experiments give you a low-resolution, blurry map of the whole thing. So what researchers did was use AF to predict the high-resolution structures of all the individual protein puzzle pieces.
And then they just computationally dock them into the blurry map.
And suddenly, you have the most complete atomic model of this gigantic machine ever created. It's the perfect synergy of AI and experimental data.
So for anyone listening who wants to get their hands dirty, the great news is that the barrier to entry has completely changed. You don't need $1 million wet lab anymore.
The new lab is the Linux terminal and some Python scripts. And the #1 tool for getting started is ColabFold.
Which basically puts AlphaFold 2 inside a free Google Colab notebook.
Right. And it has a crucial speed hack. The original AlphaFold pipeline used a slow tool to build the MSAs. ColabFold swaps that out for a much faster one called MMSS2.
And that makes a huge difference.
It makes it 10 to 20 times faster. It turns it from an overnight job into something you can do while you get a coffee.
And now with AF3 being more restricted, the open source community has stepped up again, right?
They have. MIT researchers released a model called Boltz-1. It's an open-source replication of the core ideas in AF3, the diffusion model, and the Evoformer. It keeps that next-generation technology accessible to everyone.
Okay, let's make this really actionable. If someone wants a project for this summer, what's a high-value thing they could build?
Build a utility for biologists. My favorite idea is an interaction viz dashboard. We talked about that P&Z error map.
Right, the one that shows domain flexibility.
Biologists stare at that 2D map, and then they stare at the 3D structure, and they try to connect the two in their head. It's hard.
So you build a tool that links them.
Exactly. Use something like Streamlit and Stormal. You click a blob on the 2D heat map, and it instantly highlights the corresponding flexible hinge in the 3D viewer. It would be an incredibly useful tool for any lab.
That's a fantastic idea. What about something more data mining focused?
Mine the dark matter of the genome. There are millions of predicted structures for proteins that have no known function. So you could build an orphan cluster miner.
How would that work?
You download a bunch of high-competence structures from the AFDB, you extract their basic shape features, and then you use unsupervised machine learning like k-means to group them by shape.
I see. You're looking for clusters of proteins that all fold into the same unknown shape.
Precisely. Those clusters are prime candidates for a shared, undiscovered biological function. It's pure data-driven discovery.
And for the really ambitious listener, the bleeding edge.
Protein hallucination. This is design, not prediction. You use a framework like PyTorch, and you essentially run AlphaFold in reverse. You define a shape you want.
Like a little molecular box.
And you use gradient descent to find an amino acid sequence that the model predicts will fold into that exact shape. That's the next frontier.
Amazing. And to get started, the key libraries are bio.pdb for parsing the structure files and something like py3dmol or NGLView for visualization.
Those are the essentials.
So AlphaFold has fundamentally changed biology into a data science. The challenge of finding the structure is largely solved. And now, as we're seeing, the era of designing new proteins with new functions is just getting started.
It is. And maybe one final thought to leave you with. This goes even beyond prediction and design. These new tools, like FoldSeek, which let you search databases by 3D shape instead of 1D sequence, they're uncovering incredible things.
Like what?
We're finding what are called remote homologs. These are proteins that have the exact same 3D structure and do the exact same job, but their amino acid sequences are so different that we never, ever would have known they were related.
So we're finding hidden evolutionary connections.
We're literally redrawing parts of the tree of life. We're discovering relationships that were completely invisible to science 5 years ago.
Sure.
And the tools to join in on that discovery are now available to everyone.